Hello and welcome to my channel Lab Easy. In today's video, we will be discussing a very fundamental molecular biology technique, that is isolation of plasmid DNA. Now we know that plasmid DNA are small circular double-stranded DNA molecules which are extra chromosomal DNA of bacteria, and they are capable of independent replication. Now, why do we need to isolate these plasmid DNA? Because we need to manipulate this DNA. We can insert our new genes, we can modify these genes, we can express these genes in bacteria. So because of that, the first step of these molecular biology procedure will be to isolate the plasmid DNA from the bacteria. So this plasmid DNA isolation depends on this principle that it differentially denatures the chromosomal and plasmid DNA by alkaline lysis method. So the steps briefly will be the isolation of the bacterial cells, that is the bacteria which bears this plasmid or which is growing with this plasmid. First, these cells are isolated and then they are lysed. So this lysis process takes help of SDS and NaOH. I'll be discussing these components of the lysis buffer and all later on in my video. And then following by neutralization. So NaOH is alkaline. So to neutralize this solution, we use potassium acetate. And finally, by this method, we precipitate the cellular debris, the proteins, the polysaccharides, and all other cellular components of the cell, and take the DNA in the solution. And then we use a silica membrane to bind the DNA, and then we elute the DNA using a low salt buffer or water. So now I will go detail into the steps. So the first step in this process is to prepare bacterial stock. Now the technique that I'm going to be describing here is called the Miniprep technique. There are several commercially available Miniprep kits which we can use for plasmid DNA isolation. So the first step will be to grow the bacteria on agar plates here, and these bacteria will form colonies, and we will pick a single colony with a sterile tip to use as an inoculum for 3 to 5 ml of LB culture. And in this growth medium, we will also add the specific antibiotic. So the specific antibiotic is for selection of the plasmid DNA. The plasmid DNA will contain the specific antibiotic resistance gene, by which we will be able to select the plasmid DNA, and we will be able to ensure that the bacteria will be producing this plasmid DNA. Now we transfer this culture to incubator for overnight growth at 37 degrees centigrade. And the next morning we come and we take out this culture and centrifuge to make the cell pellet. So that is how we first prepare the bacterial cell pellet. The next step will be to resuspend, lyse, and neutralize this cell pellet. So first step is resuspension of the cell. So the pellet is resuspended in a particular buffer. I will be discussing components of this buffer soon. Then we should lyse the cells. That is, we should lyse the pellets so that the cellular components now come out in the solution. And these cellular components will include everything from the proteins, from the RNA, DNA, the polysaccharides and everything. And we will be using particular reagents to deactivate or inhibit these proteins or other cellular components. And then we will neutralize the lysis. So lysis is usually done in alkaline condition. That is why this process is known as alkaline lysis. And so after that, the neutralization of the solution is also very important. So that is why we neutralize using potassium acetate. And finally, after neutralization, the cell debris are separated by centrifugation. So the cell debris will contain all the unwanted components of the cell, which will be the proteins, the polysaccharides, the genomic DNA also. But the supernatant will contain our desired plasmid DNA. So now to go through this process step by step. So first, the resuspension. The resuspension buffer contains this following components. So it contains 50 millimolar tris HCl pH 8, 10 millimolar EDTA, and 100 microgram per ml RNase A. So what is the function of these components? EDTA, we know it is a chelating agent. So once the cells are lysed, it will act as a chelating agent, and it will chelate the divalent cations, magnesium and calcium, to inhibit the proteins, to deactivate the enzymes of the cell. And RNase A will be able to degrade the RNA. Once the cell is lysed and DNA and RNA both comes into the solution, RNase A will degrade the RNA component of the cell. Now, for a 3 to 5 ml LB culture, we usually use 500 microliter of resuspension buffer, and then we resuspend the cells by pipetting in the buffer, and then we divide this solution into two tubes of 250 microliter of the solution is divided into two tubes. Now, it is followed by lysis procedure. So, in lysis, two major components of the lysis buffer are sodium dodecyl sulfate, or SDS, and NaOH. So, function of sodium dodecyl sulfate, or SDS, is to lyse the cell. That is, it dissolves the membrane phospholipids so that the cell bursts and all the proteins and DNA comes out in the solution. And when the proteins comes out in the solution, SDS also will be able to denature the cellular proteins. Now, the role of alkali is very important here, which is sodium hydroxide. And sodium hydroxide is used to denature the plasmid and the chromosomal DNA. So they are all double-stranded DNA. By the application of this alkali, we can denature them. That is, we can make them single-stranded DNA. Also, some cheotropic salts are used in lysis buffer, which because of the high concentration of these cheotropic salts, it will be further helping the DNA to bind to the silica column, that we will see in the later steps. Now, 250 microliter each of lysis buffer is added in the tubes, and then the tubes are inverted. Gently, we invert the tubes and mix for 5-6 times. So we invert the tubes and mix them thoroughly. After lysis comes the neutralization step. So lysis is done in alkaline condition. So now we need to neutralize this pH, and we use potassium acetate for that. 3 molar potassium acetate, pH 5, is used for this purpose. And the role of potassium acetate is that it renatures the DNA. So the principal role of this potassium acetate is that it helps in renaturing of the DNA. Now, when this renaturation will take place, we should remember that the genomic DNA has a much higher size. Genomic DNA is much higher in size, that is higher in molecular weight than the plasmid DNA, because plasmid DNA is a very small DNA molecule, right? Because of that, the renaturation of this genomic DNA will be much difficult, because of which genomic DNA will not be able to fully renaturate, and it will come in the precipitate. However, plasmid DNA, which is a very small DNA molecule, it will stay in the solution. So that will actually help us to isolate the plasmid DNA other than the genomic DNA. And the role of potassium acetate is also to precipitate this SDS protein complex that forms after lysis. So in this precipitate, the genomic DNA will also come, and it will separate the plasmid DNA, which will remain in the solution. So after, so actually 300 microliter of neutralization buffer is added in each tube, and then it is again gently inverted and mixed, and then kept in ice for 10 minutes. This time is allowed so that properly neutralization of the DNA can take place, so that properly the plasmid DNA is renatured. Finally, after 10 minutes, the tubes are taken out and they are centrifuged at 13,000 RPM speed for 20 minutes. This step, this centrifugation, will finally form a pellet like this. And this precipitate or this pellet will contain all the cellular debris, like the proteins, the polysaccharides, the lipids, the genomic DNA as well. And the supernatant will contain our desired plasmid DNA. So this will complete the second step. Now it's important that we have got this plasmid DNA in the solution. Now it is important to bind them to silica membrane. Now here, this step, that is the DNA binding to the matrix and finally elution of the DNA, this can also be done by another method which will involve the precipitation of the DNA by isopropanol and chloroform, and then finally washing the DNA with 70% ethanol, absolute ethanol first, and then finally washing with 70% ethanol. So this is one procedure. The other procedure is by using the kit that I was talking about, the Miniprep kits, and the kits usually come with this kind of columns. So these are the columns which will contain a silica membrane at the bottom of this column. Now we know that DNA can bind to the silica membrane. So that property of DNA we use, and we allow the DNA to bind to this silica membrane. And then we wash the DNA consecutively with mainly ethanolic buffer. And finally, elute the DNA in low salt concentration, or we can also elute in water. And finally, the DNA will come out in the solution, which we can use for further steps. So how is this binding occurs? So plasmid DNA, after the step of lysis, the lysis buffer, I told you that it will contain some cheotropic salts, and the high concentration of these cheotropic salts will destabilize the hydrogen bonding. So that the hydrogen bonding between the nucleic acid and the water is disrupted, so that the hydrogen bonding between the nucleic acid and the water is disrupted, which is why the nucleic acid, it will help the nucleic acid to bind to the silica membrane of the columns. Now, once the nucleic acid binds to the silica membrane, and then we centrifuge this column at 10,000 RPM speed for 1 minute, it will be the flow through. It will create a flow through, and the flow through will actually come out with the rest of the cellular impurities like the proteins, and polysaccharides. So the DNA will only bind to the silica membrane. However, the protein and polysaccharides and other impurities will come in the flow through. All right. So now our DNA is bound to the silica membrane. Now it's important to wash the DNA. So now we use ethanol buffer containing ethanol to wash the DNA to remove these excess salts. Because in this high salt concentration, the DNA binds to the silica membrane. Now we need to remove the salt so that we can elute the DNA out of the membrane, right? So that is why we wash the DNA with ethanol. And this we do again for 10,000 RPM for 1 minute. We use usually 750 microliter of this wash buffer. And then again, we do another round of spin, which is called an empty spin, because in this, we do not use the ethanol anymore. We put the empty columns on the centrifuge, and we centrifuge them for 11,000 RPM for 2 minutes, so that the excess ethanol can also be removed. And finally, now the DNA is bound to the column, and now it's time to elute out the DNA. So we use low ionic strength buffer or water. So usually DNA is eluted in, depending on the yield, obviously, but usually we elute DNA in 30 microliter of water. So we add this 30 microliter of water right onto the membrane, and then we centrifuge again at 10,000 RPM for 2 minutes, so that now the DNA is eluted out of the membrane, and in our append tube, we will find this 30 microliter of water, which will contain our plasmid DNA of interest. So this will complete the DNA isolation procedure. Now we will have to analyze the concentration of DNA, and for which we use a spectroscopic measurement method. So DNA gives an absorbance maximum at 260 nanometer. So we will calculate by the spectroscopy measurement, we will find out the A260, that is the absorbance at 260 nanometer of the sample. And we will also find out these absorbances, that is absorbance at 280 nanometer and absorbance at 230 nanometer. So 280 nanometer is for proteins, because proteins will give maximum absorbance maximum at 280 nanometer. So we need to check that, because we need to also check whether our DNA is contains high content of proteins or not. And 230 nanometer is for checking other impurities like phenol and the buffer components with which we wash the DNA, whether there is too much of the other components in the final DNA elute or not. So by spectroscopy, we measure these 260, 280, and 230 absorbances, and we also calculate the ratio of A260 by A280 and A260 by A230. What is the significance of this ratio? So if our A260 by 280 comes between 1.8 to 2, we can say that our DNA is a good DNA. This is because a high A260 by 280 value will indicate low content of the protein inside our DNA, which means that our DNA can be used for further molecular biology processes. And a 260 by 230 value will be like 2 or more than 2, greater than equal to 2. If the value of 260, if the ratio of 260 by 230 is greater than equal to 2, then also we will say that this DNA does not contain much contamination of phenol and all. So these ratios are important to understand the quality of the DNA that we have prepared. And also, we also need to calculate the concentration of the DNA from these the absorbance values. Now, how do we calculate that? So we know that if the DNA is double-stranded, which is the case for us also, because plasmid is double-stranded DNA at a concentration of 50 nanogram per microliter gives this A260 value of 1. So suppose we get the A260 value of our sample as 0.5. So from which we can calculate the concentration of our DNA, which will be 0.5 into 50. 50 is this conversion factor, and then we also dilute it, we also multiply it with the dilution factor. So suppose our DNA, we have eluted this 30 microliter of DNA, suppose, and we take 1 microliter of DNA and dilute it with 49 microliter of water. Okay, so therefore it means it's a 1:50 dilution, right? It is 50 times diluted. So in that case, we will have to multiply with the dilution factor as 50, and then we will be able to calculate our DNA concentration, which we will get in nanogram per microliter. Now we should also visualize our quality of DNA by agarose gel electrophoresis. Now, if we run a plasmid DNA in agarose gel, we will be able to see 3 forms of the DNA: supercoiled, linear, and the nicked form. The supercoiled form is the most dominant form of this plasmid DNA because the double-stranded circular DNA molecules mostly stay in this supercoiled form, and the supercoiled form migrates fastest in the agarose gel. So it is the lowest band that we see in the gel, followed by the linear band, which migrates slower than the supercoiled DNA, and the nicked band migrates slowest of them all. So we will see the nicked band on the top of the gel. However, if the plasmid preparation quality is very good, we will be able to see only the supercoiled form of the plasmid. And here, this is an experiment that I have performed, and I have run a 7 kilobases plasmid, and we observe the supercoiled form of the DNA. And here we cannot compare it with the DNA ladder because the DNA ladder is a linear DNA ladder. So the migration of the linear DNA ladder will not be same as that of the supercoiled plasmid, which is why the 7 kilobases band of the plasmid DNA comes lower than the 6 kilobases of the linear band because the supercoiled form always migrates faster than the linear form. However, when I used the restriction enzymes to digest this plasmid DNA, I have got these two bands of vector and my insert, which I had cloned with this plasmid DNA. And then, because of restriction digestion, these fragments are now linear fragments, and we can get an estimate of the size of the fragments by comparing with the DNA ladder. So this insert, I know that this DNA insert is around 1.7 kilobases of length, and rightly so, it forms a band in between the 2 kilobases and 1.5 kilobase DNA ladder. So that is how we plasmid the we isolate the plasmid DNA, and then we purify using the alkaline lysis method. And finally, we check the concentration of the plasmid by spectroscopy and also run the plasmid in gel to see what is the quality, how is the quality of this plasmid DNA. So thanks for watching my video. If you like, please give comments, share, and subscribe to this channel. Thank you so much.